SARS-CoV-2, the virus that causes COVID-19, is a member of the family known as coronaviruses. The surface of these viruses is covered in a membrane protein, known as spike, that helps the virus enter cells. Spike is composed of a complex of 3 identical subunits, which can be divided into 2 major sections, known as S1 and S2. The S1 section contains the receptor binding domain, or RBD, which binds to angiotensin-converting enzyme 2, or ACE2, found on cells throughout the human body. Spike RBD can exist in 2 conformations. The first, the closed or down conformation, is unable to bind to ACE2. The second, the open or up conformation, exposes the RBD and allows for binding to the ACE2. Upon binding, SARS-CoV-2 enters the cell via endocytosis and replicates. Casirivimab and Imdevimab are a pair of monoclonal antibodies that were developed to tightly bind to the spike RBD. Casirivimab and Imdevimab each attach to different and non-overlapping binding sites referred to as epitopes of the RBD without interfering with each other. The SARS-CoV-2 virion is now unable to bind to ACE2 because the bound monoclonal antibodies block the interaction between the spike RBD and ACE2. This neutralizes the virus and prevents infection of the target cell. Although SARS-CoV-2 can mutate and become more transmissible or alter the severity of the disease, only mutations in the binding sites of the antibodies can affect the ability of monoclonal antibodies to bind to spike. The likelihood of SARS-CoV-2 simultaneously mutating the 2 binding sites in the RBD recognized by each Casirivimab and Imdevimab is lower than if a single monoclonal antibody was being used. This increases the likelihood that the combination of Casirivimab and Imdevimab continues to protect against new variants of SARS-CoV-2.